First and foremost, I'd like to thank Leading Edge for this amazing opportunity. This is such a great experience and an incredible community to be a part of. And thank you again, Kara, for making all of this possible. And I'm really excited to present a subset of my work with you all today. And now just a little bit of background about myself, and my journey to where I am today has really been anything but linear. I grew up in a very small town in rural Wisconsin and went to the University of Wisconsin-Madison, where I majored in legal studies and political science, and actually didn't even set foot into a research laboratory until the summer following my senior year. And I also played on the women's ice hockey team while in college, where I won and had the privilege to win 2 NCAA Division I national championships, which is still a really surreal experience to this day. And from there, I went on to pursue both my master's and PhD in biomedical engineering with a minor in communication sciences and disorders. And I'm absolutely fascinated by the swallow, and that even the most minor perturbations to the system have severe consequences in human health and disease. And just as an example, here we have a video fluoroscopic swallow study, which is like a moving X-ray, and is the gold standard tool used in the diagnosis of swallowing disorders from a young adult individual. And you can see here the radio-opaque liquid bolus moving from the oral cavity into the pharynx and finally into the esophagus. And when we compare that to the swallow of an old individual, we can observe an age-related difference in swallowing speed, the presence of residue in the vallecula, airway invasion of that bolus, and we can also see that a second swallow is necessary to clear that bolus. And upon video fluoroscopic evaluation, this really seems rather simple. But part of my fascination as the swallowing lies in its sheer complexity, and that it requires the involvement of 6 cranial nerves and the coordination and precision of over 50 pairs of muscles to elicit. So the focus of my work during my PhD was to better understand mechanisms of diseases and disorders affecting the upper airway and how clinically relevant exercise-based rehabilitation methods affected those processes. During my doctoral work at the University of Wisconsin, I identified numerous biological processes that may underlie age-related swallowing dysfunction and degeneration, and also exercise-induced alterations of lingual and laryngeal tissues, including alterations in swallowing kinematics, swallowing biomechanics, physiology and myosin heavy chain composition, and tongue and laryngeal muscle degeneration and regeneration. And these early experiences led me to pursue my postdoctoral work at Harvard under the mentorship of Amy Wagers, where I'm now studying processes contributing to the age-related decline in head and neck tissue stem cells and mechanisms of head and neck cancer initiation to hopefully ultimately identify new therapeutic targets for age-associated diseases. And the story that I'd like to discuss with you today, and it's the story that I'm the most excited about, is related to head and neck cancer. So first, a little bit of background. Head and neck cancer is a heterogeneous group of malignancies affecting tissues of the upper aerodigestive tract, and it's the 7th most common cancer, and it accounts for more than 500,000 deaths annually. It disproportionately affects aged individuals and men. And as you can see here, incidence rates for head and neck cancers have remained relatively unchanged or even increased over the past 4 decades. You can also appreciate here as well the disproportionate rate in head and neck cancers between males, the solid filled dots, and females, open dots. And when we break down those incidence rates by anatomical location for both oral and pharyngeal and tonsillar cancers, they again have remained relatively unchanged or even increased over the past 40 years, even with reductions in smoking. And even more striking is the rate of increase over the past 20 years in tobacco and HPV-associated head and neck cancers. So primary treatments for head and neck cancer are largely ineffective and result in severe dysfunction that negatively affect quality of life. However, despite all of these, treatments have remained relatively unchanged over the past century. And some of the factors contributing to head and neck cancer are still relatively unknown and have likely hindered the innovation of novel therapeutics. Previous research has focused predominantly on the characterization of head and neck cancer tumors in patient cell lines and on outdated animal models of head and neck cancer, all of which have identified dysregulated pathways, activated oncogenes, mutations in tumor suppressor genes. But it's really clear that the current models lack in vivo biology. They're slow, they're resource intensive, and they're unfocused. And it's also clear to me that we need to have better models that more accurately model premalignancy and the inherent heterogeneity of head and neck cancers in order to discover novel therapeutics. So it's been well established in other cancer types that cancer is considered a clonal disease resulting from the progressive accumulation of mutations and the clonal expansion of a single aberrant cell or clone that ultimately leads to tumor development. And I'm also proposing that head and neck cancer may be considered a clonal disease. And to test this hypothesis, I've developed a novel preclinical model to better understand the underpinnings and dynamics of head and neck cancer. And hopefully that will help us identify for the first time the cells of origins of various head and neck cancer subtypes. So numerous groups have postulated that cancer risk increases with the total number of stem cell divisions over one's lifetime, and that on average 3 mutations occur each time an adult somatic tissue stem cell divides. And this acquisition and accumulation of mutations of head and neck tissue stem cells could also be a likely mechanism driving head and neck cancer initiation. So how exactly am I proposing to study this? We're using 2 different models to understand the clonal dynamics and molecular underpinnings of head and neck cancer initiation. And the first model is related to a tobacco-induced carcinogenesis and the use of a tobacco mimetic 4NQO to induce DNA adducts into various head and neck tissues and their resident stem cells. And when we examine the interaction of time, age, and sex with carcinogenesis, strikingly, this model of tobacco-associated head and neck cancer recapitulates in mice the sex and age variant pattern seen in humans, with higher prevalence of gross lesions in older animals and in males versus females. And in the study, we also observed the age-related loss of head and neck tissue stem cells that is also exacerbated by carcinogen exposure, and even tobacco-accelerated clonal expansion of epithelial stem cells with age. Our preliminary data also suggests epigenetic and transcriptomic changes that are implicated in key biological processes, including oncogenesis, epithelial to mesenchymal transition, and stem cell differentiation. So in this second model, I'm taking a forward genetics approach to model head and neck cancer initiation by targeting AAV CRISPR gene editing to different somatic tissue stem cells. So I generated an AAV single guide RNA library targeting the 20 most frequently mutated head and neck cancer genes across an array of cancer types and tissues. And I'm locally injecting that AAV machinery into the head and neck tissues of Cas9 mice with Cre-driven stem cell specific promoters that then allows us to restrict that editing to our cell types of interest. So first, what is AAV? It's a non-envelope virus that affects dividing and non-dividing cells, and it's able to harness and hijack endogenous cellular repair mechanisms to either achieve gene disruption or gene repair. And why AAV? Because it's highly customizable for precision-based gene editing and gene therapies. It has a large number of serotypes for the targeted editing and transduction of different cell and tissue types. It's capable of driving very robust transgene expression. It's safe, and it's been used in numerous clinical trials. First, we perform serotype testing across our different head and neck tissues and their resident stem cells. And this data shows the effect of transduction or infection of multiple stem cell lineages, specifically muscle stem cells, mesodermal progenitors by the AAV9 serotype that is represented in neon green, and epithelial stem cells by the AAV6 serotype in blue at 2 different doses. And also the high and robust gene editing rates or the frequency of genomic insertions or deletions, indels in our stem cell populations and tissue types of interest. So then at that point, we were ready to inject our library and our different serotypes that we identified. And at 10 weeks post-injection, we observed neoplastic tongue lesions in all injected animals. And the development of tongue tumors at different rates depending on the cells of origin that were targeted. And by 20 weeks post-injection, all mice that were injected had developed tumors. And as you can see here, the tumors were incredibly heterogeneous, anatomically and morphologically, depending on the target tissue and the cell type. And here we have an additional slide just showing some of that additional heterogeneity across additional tissues. And just as exciting, we were able to show that our AAV-induced tumors generated in the mouse were able to recapitulate the heterogeneity of human head and neck cancer tumors, with different types of mutations being detected in multiple CRISPR-targeted candidate head and neck cancer driver genes. So now I've shown you a new mouse model that I've generated to study human-associated head and neck cancers, and our preliminary data suggesting that head and neck cancer is in fact a clonal disease and can be initiated by both a generation of mutations in resident tissue stem cells and in the whole tissue itself. And again, another summary slide, we observed that head and neck tissue stem cell frequency and epigenetic and transcriptomic modifications are differentially impacted by age and following the exposure to a tobacco mimetic, and that we can use an AAV-driven oncogenesis model to drive and better understand mechanisms of head and neck cancer initiation. And in the future, I am planning to continue this work where I'm going to be studying mechanisms of age-related decline and head and neck cancer initiation using novel models of human disease to hopefully identify novel therapeutic targets across age-associated diseases and cancer. And at that point, I just would like to thank you all again for this opportunity. This has been a fantastic community to be a part of. Again, thank you again to Kara. And I'd also like to acknowledge my funding sources.